So I would like to welcome you all to today's journal club, which is organized by the EASD Academy in collaboration with Diabetologia. And so it's my pleasure to welcome Joanne Boldison, who will present her work, which was published in Diabetologia. And so my name is Claudia Cavalti, and I'm also a member of the EASD Academy, together with Sandra Smink, who is my co-host today. And so how we do it, I will shortly introduce Joanne, and then we go over to her presentation. And in the end, we have time to ask some questions. And it's also important to us that we ask, not only we can ask questions regarding her work and the paper, but you're also invited to ask some career-related questions, as Joanne has now started her independent journey in research. And I think this is a very crucial moment of everyone's career. So I would like to thank you, Joanne, that you are here today with us and will present your work. So Joanne did her PhD at the University of Bristol, and she worked there on the topic of ocular autoimmunity. Then she did a postdoc at Cardiff University together with Professor Susan Wong at the Diabetes Research Group, and was studying the immune pathology of type 1 diabetes. And now, since 2020, she became an independent research fellow at the University of Exeter and is still interested in the role of B cells and type 1 diabetes pathogenesis. So thanks so much, Joanne, and over to you.
Thank you. For the lovely introduction, and thank you for the invite today to present my work that was published in Diabetologia and to present here at the EASD Academy Journal Club. So I thought I'd just jump straight in and give you a brief introduction and highlight some stuff before I talk about the data. So I don't think with this audience, type 1 diabetes needs much introduction, but if people don't know, it's an autoimmune disease of the pancreas where you get a destruction of your insulin-producing beta cells and a loss of blood glucose control. And I just wanted to really highlight before I talk about the paper, the importance of studying B cells in type 1 diabetes. And this has really been highlighted by a couple of studies, in particular the rituximab trial, so anti-CD20 treatment a few years ago now, and also the really exciting data that came out from my colleagues at Exeter with Dr. Pierre Leet and Sarah Richardson, showing that there was an abundance of CD20 positive B cells in pancreatic islets from donors that were diagnosed young, and it's associated with a more aggressive form. And but just to sort of switch then really quickly, because I know I've only got 10 minutes to squeeze everything in. This particular study, this particular paper that was published uses the NOD mouse model, which is the non-obese diabetic mouse model. For those of you who are not familiar, these mice develop spontaneous diabetes around 18 to 19 weeks. And in females anyway, have about 80% incidence. This can vary depending on the colony you're in, but everything that I'm going to talk about onwards has been done in the NOD pancreas. So I just thought I'd sort of set the scene really about where the project came from. And when I was at Cardiff University working with Professor Susan Wong, we were interested in insulin-specific B cells, so autoreactive B cells, and the effects of anti-CD20 treatment. And at the time, we were phenotyping B cells in the NOD pancreas and looking at these insulin-specific B cells. And we decided on a gating strategy of IgD or CD19 against CD138. And we sort of compartmentalize these B cell subsets. And what we found was when we were phenotyping the insulin-specific B cells, we found that in these particular subsets that expressed CD138, they were enriched. So these subsets expressing in the blue gate and the grey gate were enriched in autoreactivity. And then we did a whole body of work, including kinetics and having a look at the progression of diabetes in the pancreas and the abundance of these subsets, and basically found that the double positive subsets were more abundant as disease progressed, and they became more of a towards a 1-to-1 ratio in comparisons to their CD138 negative counterparts, so the B cells in the green gate. So just to highlight then that these 60 to 20 weeks old is everything I'm going to talk about in terms of the new data and the gene expression profiling that we did. But so this particular project came up because of the observation just that we'd seen here. And so I was really interested in trying to understand if these B cells had a different function in the pancreas, and if there was a way of being able to pull them out more efficiently, whether we could identify them more efficiently and maybe even target them. And so I asked if they had a different gene expression profile. And this was a grant that was submitted to Diabetes UK as an early career award and was successfully funded. And so we went about doing the experiments, and we took groups of mice that were 16 to 20 weeks old, isolated the pancreatic islets and the pancreatic lymph nodes, and then FACS sorted as our gating strategy that we'd identified earlier, and did the RNA isolation and the gene array. And I think what was quite surprising and maybe slightly disappointing to me was the fact that they had very similar transcriptional profiles in both the pancreatic lymph node and the pancreatic islets. And in fact, the only gene that was significant to a corrected p-value of less than 0.05 was Syndecan1, which encodes CD138. So this was quite surprising, but when we mined the data further, and I should say, sorry, before we go further, in the paper, we also studied the CD138 population that has lost IgD and has a low expression of CD19, but I don't think I've got time to cover that in my talk. So that is in the paper though, and I think it's figure 2 and in the supplementary data. And I'm happy to answer questions on it. I just didn't think I'd have enough time to cover everything. And so when we looked at the data further, I think it was really apparent how different the genes were expressed between the tissues. And so instead of comparing this way, we also compared this way in both subsets. So both the CD138 negative subset and the CD138 positive subset. And when we put the Venn diagram together, we found that we have 388 differentially expressed genes in that were shared between the single positive and the double positive subset. But what was really interesting was the fact that we had also unique genes to each subset, and quite a few as well. But in terms of thinking about the shared genes of the two subsets, just to point out that we found an upregulation in PRDM1 and IRF4, which is associated with the plasma cell differentiation pathway. And surprisingly, we found genes downregulated that were associated with antigen presenting. And I'll cover a bit more about the other genes that we found upregulated in the next slide. But what I think was really obvious to show between the tissues was when we did the principal component analysis, and we could see that we had, the PLN B cells really bunched together, and then a slightly further spread for the pancreatic islets, but you can really see the transcriptional distance between the two tissues, which was really interesting. And so we went one step further and did some pathway analysis and gene ontology analysis. And so we used IPA and various web tools, which is described in the paper. And we found that there were various activated pathways in both gene sets. So this is a Z-score that IPA gives you to show that, if it's red, it's activated. If it's deactivated, it's blue. And in the graph here, you can see the significant p-values for each subset that corresponds to that particular pathway. And so we found that interferon signaling was upregulated, pattern recognition receptors, and B cell signaling pathways that are very similar in SLE to type 1 diabetes. And so when we did the IPA analysis and the gene ontology analysis, we combined the genes that we found that were involved in those pathways that were differentially expressed in our subset and created a sort of gene signature heat map, if you will, for both subsets. So here on the left in the green here, you can see that the pancreatic islets, all these genes were upregulated, and the same in the double positive B cells. And you can see that upregulated were certainly certain cytokines and lots of interferon stimulated genes. The gap in the heat map here at the bottom is these were 3 genes that were only upregulated in the single positive cell subset, and the 2 genes here in the double positive cell subset, if that makes sense. But one of the upregulated genes that was of interest was TLR7, knowing the literature associated with autoimmunity and TLR7, and the fact that it was quite highly upregulated. So I chose TLR7 to confirm by protein expression first, and I did this by confocal microscopy. So this is just one picture of a pancreatic islet in a NOD mouse that was diabetic, and there's more pictures in the paper in the last figure that you can go and look at. And I'm hoping that this translates on screen, and you can see that the TLR7 expression in green and the CD20 positive B cells in red, and you can see that we've got double positive B cells in the pancreatic islet. So those B cells are expressing the TLR7 protein. And then we've also followed this up with with flow cytometry, and using our representative gates. And we could show that in the pancreatic islets, both B cell subsets, so the same as our gene array, were upregulated with TLR7. But what was really interesting was that it looked to me, and significantly, that those double positive B cells were expressing TLR7 more readily, the protein. And so I thought that was really exciting, and have gone on to do some further work that's not in the paper. So hopefully watch this space. And just to whistle stop tour, I know, sorry, to put the graphical abstract as the summary, just to show sort of the key differences between the tissues, and the fact that these B cells, once they arrive in the pancreas, they're undergoing these changes because they are educated by their environment. And that happens in both B cell subsets, whether they're autoreactive or not. And that environment is driving genes that are associated with antibody secretion, inflammatory cytokines, TLR7, and downregulation of antigen presentation. And the one thing is in the paper that I haven't touched on because I didn't think I had time, but was the fact that in the double positive B cells, there was some genes that were only expressed in that gene set that were associated with late cell cycle genes and some growth factors. And I think that that's in figure 5 of the paper, if anybody wants to go and have a look at it. And so lastly, I'd like to thank everybody, specifically Professor Wong at Cardiff University and the Exeter team, in particular Jess and Pierre, who were very helpful in doing confocal microscopy in a new environment when I transferred to Exeter, and Diabetes UK, which this grant funded 95% of the project, so it wouldn't have been possible without them. And I'm happy to answer any questions.
Wonderful. Thank you so much, Joanne. That was really very exciting and interesting. And so I want to open the round for questions, and you can raise your hand. You can also write into the chat, but it's always nicer to ask directly. So if you want to open your camera, that would be really preferred. So maybe I'll start in the meantime. What I found really fascinating is that it seems that the B cells, they acquire like an innate immune signature. Is that right? And is this something, you know, observed in other disease conditions? And if that's the case, what other disease conditions do have kind of a similar innate immune signature in B cells?
Yeah, so certainly the other diseases you can see on the pathway SLE came up, and there's been a huge body of work in SLE about the interferon signature in B cells as well as, you know, sort of as in general, and which has shown some sort of cooperation of TLR7 as well with the B cell receptor in terms of, in SLE anyway, encouraging those autoreactive antibody secreting cells that do the damage in SLE. So yeah, certainly in SLE and in RA as well, I think.
Great, thanks. Some other questions came up. So Sandra, I'll start with you.
Yes, thank you. Very interesting talk and interesting paper, I think. And I had maybe a bit more technical question about because you did the PLNs and the islets, but did you also look into maybe other tissue or other lymph nodes or other organs to the B cell population there? Will you expect that it's the same for as in the PLNs?
Yeah, good question. So at the time, I did fact sort when I was doing the experiments. I did sort the populations from the spleen, but I was, you know, sort of very limited in funds, so couldn't actually afford to run those spleen cells, and chose the pancreatic islets and the pancreatic lymph node. But the plan is to follow up with maybe some sort of directed qPCR and having a look at those genes that we found, you know, that we've been able to pull out, you know, in the pancreatic lymph nodes and islets and the changes, to go back and have a look at those samples. I have done some other functional experiments sort of further on from the paper, from the splenocytes, looking at the different subsets and how they respond to different stimuli. So yes, it would have been fantastic to be able to have an array of different lymph nodes from the animal, but it just quite wasn't possible to fit everything in.
But so with your experience, and would you expect that they will be different if it's further away? Because I can imagine that the pancreas, the autoimmune reaction going on in the pancreas influence the PLNs still, but do you expect it will be different in the other?
So they certainly are different in their functionality and what they express after you've treated them with, say, interferon alpha. Okay. But I haven't found, you know, one particular marker that is, you know, sort of really different in terms of pulling them out ex vivo and without giving them stimuli in the spleen anyway, which is sort of one of the difficulties I had originally. It seemed that CD138 was really the only difference in the marker, and that came out in the gene array as well as the previous flow cytometry I'd done in the Diabetologia paper in 2019. So it seems that CD138 seems to be the thing that changes in these cells, or at least the way that you can mark the subsets.
Okay, thank you.
Any questions from the audience? Otherwise, I'll have one question. So you showed that, or sorry, Pia, I saw that you raised your hand previously. Do you have a question?
I did, and then I thought I could. I'm lucky enough to work with Jo, so I thought I might ask the question in private, but I'm going to go for it anyway.
Great.
I'm always curious about that idea of antigen presentation and where it happens. So I've had, you know, taken some images that look like B cells are interacting with T cells at the islet site, but your data suggests that the antigen presentation genes goes down in the islet. but I'm guessing that this is at a particular stage of diabetes, so rather than in the kind of initiation phase, because I think as far as I understand with mice, the islets are all inflamed simultaneously, whereas in the human pancreas, it's kind of a much more lobular disease. Is that right? And so is there any way of staging that kind of destructive process in terms of the antigen presentation?
Just to clarify about the mouse human thing, the islets are different in terms of how fast they are destroyed. They don't all go at once.
Oh, okay.
You sometimes, and not every mouse is the same either. There is some heterogeneity about how fast some mice go, and some don't, and some don't ever develop diabetes, and have islets remaining, with only a few islets that have few insulin secreting cells. So in terms of that, yes, I mean, obviously human and mice, they're different, but in terms of the mouse, no, they don't all go at the same rate.
So you could stage that in some way?
You could, in terms of looking at individual islets, I think you probably could. You could probably do individual islets looking at the different stages of, sort of whether it's early or late or later. It's very difficult to isolate islets when mice have become diabetic and they've clinically sort of become diabetic, because you're limited to number one, how long you can keep them, but also the fact that lots of your islets are destroyed, and so there's nothing to isolate. So that does limit in terms of how you approach that kind of study, and whether you'd be doing some sort of confocal microscopy and sort of trying to do serial sections in terms of looking at antigen presentation in that respect.
Okay, thanks.
I hope that answers your question, Pierre.
I'm sure I'll come back to you thinking about it more and more. It's a mither of mine.
Wonderful. James, you have a question.
Yeah, thank you very much, Joe. Really good talk. My question, I guess, is related to how in your microarray data you picked up that you've got a similar gene signature to SLE, potentially because of TLR7 and those other signatures. But I'm just wondering, is there any other data in other autoimmune diseases where there is this increased or population, and whether it's increased in those autoimmune diseases at all? Increased
population
of CD138 IgD positive or CD138 negative, if they're altered in other autoimmune diseases too?
So in terms of mice, there have been one, but to the best of my knowledge, one other study that was looking at those CD138 positive B cells that were enriched in autoreactivity, and that was in a lupus model of a mouse model. But in terms of the other literature that's out there, it's quite limited in terms of this particular cell subset from what I gather, which is why I was sort of desperately trying to find a marker to pull them out and to study them, and to try and find out more about them, because so I mean, it seems that there's not a huge amount from the best of my knowledge, but there is the one lupus model paper.
I have another question regarding the point where you mentioned that the environment, kind of the lymph nodes versus the pancreas, shapes these types of immune subsets. So this was now what you've shown is in the NOD mouse, right? So would they be also, would they also exist like this in a healthy mouse, or is it just because of the autoimmune disease that they kind of have these two populations, and in a healthy mouse, would they also be so different?
So when you say a healthy mouse, I would assume that you mean a mouse that's not susceptible to autoimmunity. Is that what you're asking?
Yeah.
Okay. So
because these mice, they were all diabetic, or they were just no,
they're not diabetic. They're non-diabetic, but they're at the stage where they have a lot of pancreatic inflammation in terms of
Okay.
you expect them to be going diabetic because the mice that I actually took for these experiments were not diabetic, no.
Okay, that's important. Yeah. And let's say you would look at black 6 mice, regular kind of non-susceptible mice.
Yeah, I would guess that certainly, of course, you wouldn't have them in the pancreas because you wouldn't have the inflammation because it's wouldn't be there. But certainly in the spleen, if I had to guess, I would say that the population would be there because, you know, naturally you have autoreactive cells in humans as in healthy people, as well as you know, sort of people with whether they're susceptible to autoimmunity or not. But I would imagine that maybe that particular CD138 subset is maybe smaller in compartment, or is maybe controlled better in terms of their peripheral tolerance or central tolerance in those healthy mice. So yeah, you'd have to only obviously ask the question in terms of peripheral.
Yeah, got it. Yeah, that makes really sense. Just another quick technical questions regarding that, because we were also trying to isolate immune cells from the islets or for the pancreas, and as you mentioned, it's a rare kind of, you know, it's not a lot, right? And you write in the method that you pool mice. So how many mice did you have to pool for one sample to get a kind of
7
data?
to 8.
7 to 8 mice is one data point.
Yeah.
Okay, that's interesting.
Yes, 7 to 8. Yeah, to be able to have enough of each population to be able to get, you know, enough RNA from the FACS sort. Yeah, so 7 to 8 mice each time. Yeah.
Okay, great. Thanks. Sandra, over to you.
Yeah. So I was thinking a little bit about translation towards humans, as was talked about a little bit before. So how easily can this, or is there already something known about the B cell population in humans? How is it? So is it one-to-one translatable, your mouse data to?
Yes. Yes, it is. Pia's going to be smiling behind her camera at the moment, because we've been working on trying to translate some of these proteins and genes that were in the mouse array that were upregulated into looking at using the Exeter Biobank in collaboration with Sarah Richardson and Pia at Exeter, and that's just been funded by JDRF. So, and we've had some really exciting preliminary results showing that can definitely see some positive B cells that have got some MXA and possibly some TLR7 too. So that's in terms of the pancreas, in sort of translating those proteins to see if the B cells in the pancreas also have, you know, take up that interferon signature that they're sort of surrounded by. And in terms of the peripheral blood B cells, there is some really exciting data in SLE about a population of B cells, or DN2s, or activated naive B cells, that seem to have TLR7 hyper-responsiveness. So I've been trying to have a look to see if those B cells in the peripheral blood of donors, individuals with type 1 diabetes, we have some elevated numbers of B cells that might be TLR7 hyper-responsive.
Cool.
So as we speak.
So in the paper you also mentioned like some that it opens up some potential therapeutic avenues, and I think you mentioned TLR7 as a potential therapeutic target. But can you maybe speculate a bit on how you imagine this in type 1 diabetic patients? What will it mean for a patient?
Yes, so I think it's important to note here that I think that there's a point where we have to understand more about the donors that or the individuals that you would be wanting to treat. So thinking about obviously delaying onset of diabetes, so treating earlier, so understanding the effects in, of anti-TLR7 or B cell intervention, which, however it is, that earlier on would obviously be better, but we have to also think about whether that's adult or juvenile, and especially in the context of Exeter's, Pia and Sarah's fantastic data, thinking about the different endotypes, and whether it might be that something like, TLR7 intervention or B cell intervention might favour the younger people who have a younger age of onset, but also thinking about people who already have diabetes, thinking about the damage has already been done, as it were, and obviously you'd be coming in too late. But I think, and I was thinking about this the other day, is that trying to think about if individuals would have islet replacement therapy, it might be that this actually might be useful in terms of trying to halt that recurrent autoimmune attack after people have had islet replacement therapy or something like that. So for those people who've already got type 1 diabetes and are clinically diagnosed.
Oh, very, very interesting. Thank you.
So maybe I'll switch to a career-related question then. So you told us just before we started at 5 that when you moved to the University of Exeter and started your own lab, that was right at the beginning of the pandemic. So I guess that must have been a big challenge. But other than that, what do you think is what was the biggest, or still is the biggest challenge for you in transitioning kind of towards becoming an independent researcher and having your own group?
Well, the biggest challenge,
or just a challenge,
challenge. Yeah. It's all a challenge, isn't it? I think yes, it's been difficult transitioning, especially during a global pandemic. Everybody's been fantastic about it though, and it's not just me that had to transition. I'm aware of that. I think at the moment, really, the biggest challenge for me personally is having enough hands, or getting enough larger grants that you can apply for that has a postdoc salary, a PhD student salary, or a technician salary for you, because currently it's just me. So, I am only one person, and people can only, sort of help me so much. Everybody has their own research to do. So I think that's this particular point in time, that's the challenge, is being able to sort of carry on all the projects that I really want to carry on with, and be able to sort of write the grants in this, that, and the other, but to sort of, obviously you can't spread yourself too thin, or try not to anyway.
And what would be your advice to younger kind of early career researchers who envision also to move in an independent direction? So what do you think is an important advice?
I think the one thing that I think would have been helpful for me earlier on was understanding what pots of money were available to sort of small projects that you would be able to build your CV. During my PhD, I don't know if that was even spoken about. It just wasn't really sort of, I don't know if it wasn't encouraged, or whether I didn't reach out enough, because you're quite sort of bogged down with a thesis and doing a PhD. But I think that would be my advice, is to find out what other sort of little projects that you could do, little pots of money, whether that be engagement, whether that be a method you want to set up. It all sort of adds to your CV that shows that you're sort of dedicated and sort of you're sort of proactive in terms of sort of trying to find that sort of funding for independence. But I think that for me, that would have helped me maybe in my CV and my sort of, whether that was grant record, publication record, if I'd have done that earlier.
That's actually an interesting point, because I feel like, you know, kind of also as a young PI, you always feel that you don't want to overload your students. So kind of they are already working a lot and on the project. So kind of getting their own money or doing some grant writing. But it's true. I mean, you can also see it from that side, that it really, that as a student you can also grow in this this process, and maybe we as a PI can really encourage people to, even in these early stages, to apply for money. And yeah, that's a very good point.
Yeah, it's just, you know, anything, I think, you know, or at least being, you know, sort of aware of when you can apply for money, I suppose, you know, those smaller pots, whether that's within your institution or you know, a smaller early career award, and I just, it was definitely something that was missing for me, you know, maybe because I was too overwhelmed with lots of nice work at the University of Bristol, but I think that would have definitely helped me and my CV, and maybe it's not such an issue now, because a lot of the sort of postdoctoral years where you're applying for funding have quite a lot of funding bodies have dropped that, but I was finding myself really sort of cut off, that's too junior, that's too senior, and I wasn't sort of you know, I was sort of finding myself very, very limited of what I could apply for.
Yeah, that's very good. And I see, Pia, you also had a comment on that in the chat, that it was great also be a co-writer in grants.
So,
Yes.
yeah, even within my PhD, my supervisors were sort of encouraging me when there were findings that were associated with my work to take part in that writing process. Although I wasn't writing the big grants, I was reading them, I was listening to the processes of what they were doing and they had to do. So it was kind of an insight into what was going on in the background as well, which I think was really valuable.
Yeah, I mean, I was in the grant writing process as soon as I'd finished my PhD and going for fellowships and other stuff. But it wasn't until I got to Professor Wong's lab that it was sort of made apparent to me that there were smaller pots of money that you could have access to, and I think that would have been helpful earlier on, even in the last year of my PhD. That would have been, you know, sort of definitely helpful earlier.
Great. Thank you so much. I really enjoyed it a lot. I don't know whether there are additional questions. Sandra, you still have something, or should we close the session?
No, I don't have any questions anymore so thank you for the presentation, but I do want to say some other things to the audience, is that we, as the Academy, as the EASD Academy, will be looking for some new members. So there is some advertisement or what we are looking for on our website. So you can apply if you are interested to join us. And the other thing I would like to announce is that our next webinar will be on the 9th of March, and keep posted for the newsletter or the website for exact details on that and for registration to the webinar.
Wonderful. Thank you so much, everyone, and especially to Joanne for this great talk and also the question round. And have a great evening.
Thank you. Thanks for inviting me. Bye, everybody.
Bye, everyone.
Thank you.